Hello students, welcome to Shri classes. We will begin with chapter number 9, Heredity and Evolution, chapter flow. The chapter has been divided into two parts. One is heredity and next is evolution. Heredity, in this we are going to study the mechanism by which variations are created and the variations are inherited from generation to generations. In the evolution part, the long-term consequences of the accumulation of variations. But anyway, for the current syllabus, this evolution part has not been retained, or evolution part has been deleted from the current syllabus. Hence, we will not be discussing the evolution part, only the heredity part, and majorly the heredity part consists of Mendel's monohybrid experiment and dihybrid experiment, along with that a sex determination. These are the parts that we are going to study and discuss. So let us start the chapter now. So what is heredity here? Heredity refers to the transfer of characters or traits from the parents to their offsprings. So the transfer of characters or traits from the parents to their offsprings is called heredity. So here, what do you mean by characters? Characters means for example, the visible features, I will say the characters. Characters generally refer to the visible features. If you consider the human beings, of course, all the human beings will have two legs, two hands, etc. The body structure will be similar, but definitely there are certain features by which we would be able to distinguish from persons to persons. So those visible features which enable us to distinguish between two persons or the two organisms of the same species is called the characters basically. So these characters are transferred from parents to the offsprings, and the process is called, and that transfer of characters is called the heredity. So what are variations? The difference in the characteristics or traits among the individuals of the same species are called variations. Example, we'll take over here, the dogs are there, here children, and grapes, beetles. What is that enables us to distinguish between the grapes here? The color. So color is a variation in this category. What about here? The red beetles and the green beetle. Again the color here. With respect to children, even though the body structure is similar, there are many features by which you will be able to distinguish from one child to another child. Those are called variations. Here also the dogs, the colors basically are the ones by which you will be able to distinguish between two dogs. So variations are the differences in the characteristics. Characteristics means visible features. So the differences in the visible features are called the variations. And what is the importance of variation? They enable organisms to adapt themselves in a changing environment. Many times the variations enable organisms to sustain harsh environmental conditions. Sometimes if the organisms are not able to sustain the harsh environmental conditions, then they will die or on the verge of extinction. Also, the variations form the basis for heredity, evolution and development of new species. So this is the importance of variation. One is the organisms adapt themselves in a changing environment to sustain the life, and second thing is variations form the basis for heredity, evolution and also the development of new species. So I hope you have understood what is heredity and what are variations and what is the importance or the significance of a variation. Next, accumulation of variations during reproduction. If you consider the accumulation of variations during the sexual reproduction, we have to study in variation in asexual reproduction and variation in sexual reproduction. What do you think? in asexual reproduction the variations are more, or in sexual reproduction the variations are more? What is that your guess? See, variation in asexual reproduction, only one parent is involved. Why the variations occur? Because no biochemical reaction or no biochemical process is 100% accurate. There exists variations in the biochemical reactions that produce the DNA copy, and as the DNA copy, as there exists variations in the DNA copy, the blueprint differs and the body structure differs. So if only one parent is involved, is the variation more? Or if two parents are involved, is the variation more? If two parents are involved, the variations will be definitely more. The variations, accumulated variations of parent 1 plus combining with accumulated variations of parent 2 lead to the more number of variations in the offsprings produced by sexual reproduction. So let us see some of the features of these two, variations in asexual and sexual reproduction. Let's see that. So variation in asexual reproduction, when organisms reproduce, the offsprings show variations due to inaccuracies in DNA copying. This we have studied already. And these variations are less in asexual reproduction because only one parent is involved. So organisms tend to preserve similarities with very minor differences between them. If there are minor differences, then I can say in the offsprings that are produced by asexual reproduction, the offsprings can be identical. That means very less differences will be there. Offsprings can be identical. One example here it is given, bacteria. If one bacteria divides into two and another, those two bacteria once again they divide into 2 and 2. So one bacteria dividing into 4 individuals, almost all these bacteria look alike only, almost identical. Very little variations will be there. Probably that little variations may help the bacteria to sustain some very harsh environmental conditions such as high temperatures or very low temperatures. Like the bacteria can sustain very high temperatures in the deserts, and also bacteria can live very well in Antarcticas and also which are sub-zero temperatures. So the variations, of course the body structure and all of the bacteria will be similar. Almost most of the time, most of these bacteria will be similar in nature, or I can say identical, and very less variations can be observed in bacteria. And therefore the organisms produced by asexual reproduction are said to be identical. What about variation in sexual reproduction? In sexual reproduction, as there are two parents involved, the inaccuracies in DNA copying is more, that leads to greater diversity or variations in the offsprings generated, because the variations of the mother and the variations of the father both put together, more variations can be expected in the offsprings, and hence we can say the offsprings are similar but not identical. That you should keep it in mind. So offsprings produced by sexual reproduction are similar but not identical, whereas in asexual reproduction we can see the organisms of the offsprings produced are little identical. And of course, as I said, these variations sometimes they are helpful to sustain the harsh environmental conditions. Sometimes these variations are not helpful. In that case, the organisms may die and become extinct. So in this slide, I hope you have understood the nature of variation in asexual reproduction and sexual reproduction. Next, what are inherited traits? The traits or the characteristics. See here, characteristics or the traits are same, one and same. Traits means the visible features. Characteristics means also the visible features. So both are same. So inherited traits are the characteristics that are transferred from parent to the offsprings from generation to generation. That is called inherited traits. And thus inherited trait is genetically determined characteristic that distinguishes a person. So inherited traits are genetically determined. Please remember, inherited traits are genetically determined. It depends upon the genes of the mother and genes of the father. So the inherited trait is a dependent factor of genetically, it is dependent upon the genetics or the genetically determined character, you can say, the inherited trait. Now, next is the rules for the inheritance of traits. The rules of inheritance, the rules for the inheritance of traits was formulated by Gregor Mendel, who was a mathematician, but he did the experiments on peas plants, and finally he concluded certain points or the rules. He finally devised the rules of inheritance. He determined the rules of inheritance. So more famously, these experiments are known as Monohybrid Experiment and Dihybrid Experiment, and that led to the field called genetics. And Gregor Mendel is also called the father of genetics. So let us see what are the points here. Characters are transferred through genes present in the DNA molecules, and DNA molecules are present in the chromosomes, and chromosomes are present in the nucleus of the cell. So basically, characters are transferred through genes from parents to their offsprings. The inheritance of characters is due to the fact that both father and mother contribute equal amount of genetic material to the child. Is it not? The inheritance of characters is due to contribution by both mother and father of the genetic material in equal quantities. Okay? So for each trait or a character, there are two factors, or also you can say two genes or copies of the genes, one from the father and one from the mother. So Gregor Johann Mendel conducted experiments with garden pea plants and determined the rules for the inheritance of traits. See, when we talk about the traits here, a couple of diagrams are given. Free earlobes and attached earlobes. Here, these are the free earlobes, and here it is an attached earlobe, you can see here. Okay, and this is also due to inheritance. Maybe a father could be having an attached earlobe, and therefore the child could have an attached earlobe, something like that. So basically, Mendel's contribution is that Mendel, he devised or he determined the rules for inheritance of the traits. Next. So a small note is there here based on the in your textbook it is given. Let us go through this one. Gregor Johann Mendel, 1822 to 1884. Mendel was educated in a monastery and went on to study science and mathematics at the University of Vienna. Failure in the examinations for a teaching certificate did not suppress his zeal for scientific quest. Most important, this sentence. He went back to his monastery and started growing peas. Many others had studied the inheritance of traits in peas and other organisms earlier, but Mendel blended his knowledge of science and mathematics and was the first one to keep count of individuals exhibiting a particular trait in each generation, and this helped him to arrive at the laws of inheritance. So remember, the laws of inheritance were discovered by Gregor Johann Mendel. Next, Mendel's experiments. Mendel used a number of contrasting visible characters of garden pea plants, such as round and wrinkled seeds, tall and short plants, white and violet flowers, and so on. So in pea plants there are many varieties. With respect to seeds, the color can be green or yellow. With respect to shape, round and wrinkled. With respect to height of the plant, tall and short plants. With respect to color of the petals of the flowers, white and violet flowers, and so on. So he performed his experiments on pea plants. Gregor Mendel is called the father of genetics. His experiments give the information about mechanism of inheritance of traits from one generation to another generation. Finally, we are going to conclude what is the mechanism of inheritance, and Gregor Mendel's experiment have led to the mechanism of inheritance of the traits from one generation to another generation. Question is, why did he select pea plants for his experiments? Generally, this question appears in the exam. They have a shorter life cycle of 3 months. Within 3 months, a pea plant can be grown right from the sowing of the seeds to get the matured plant and to get the peas. So they have a shorter life cycle of 3 months. They produce more number of seeds and can be grown easily and can be handled very easily. And pea plants have more number of contrasting visible characters. That is one of the important factors. And they produce bisexual flowers where artificial cross pollination can be carried out easily. See, most of the times pea plants are produced by self pollination, and generally the tall plants will produce only tall plants, short plants will produce only short plants. But it is very easy to cross pollinate also and check the results. And this is why Mendel selected the pea plants. So they produce bisexual flowers where artificial cross pollination can be carried out very easily. So there are two experiments with respect to Mendel's experiment that we are going to study. The first experiment is called a monohybrid experiment, and second experiment is called a dihybrid experiment. What is this monohybrid experiment is used for? It shows the dominance of traits or expression of traits. What is the meaning of expression of traits? If a particular trait is expressed or dominated, we say that trait is dominated if that is a predominant visible character. For example, shortness and tallness are the characters. If we observe the tallness in most of the plants, then we say the dominant trait is a tallness. If we observe shortness as in more number of plants, then we can say the shortness is the dominant trait. So dominant or expression of the trait means the characteristic that has got expressed, the characteristics that becomes the visible feature of a particular organism. Here with respect to plant you can say. And in the dominance of traits or expression of the traits, which is the monohybrid experiment, what is the meaning of this monohybrid experiment? Mono refers to single. So one pair of contracting characters were compared in the monohybrid experiment. The cross between two pea plants differing in one pair of contrasting traits is called a monohybrid cross. One pair of contrasting traits. One pair means what? I can take the shape. Shape is one trait. In that one, the round and wrinkled. Round is one character, wrinkled is another character. Or short and tall and short is another one pair of contrasting characters. Color of the petals, violet or white or violet forms one pair of characters. So round and wrinkled forms one pair. Green colored seeds, yellow colored seeds also form one pair. Tall or short plants also form one pair. Violet or white flowers also form one pair. So in an experiment where only one pair of contrasting traits means opposing traits. Contrasting means opposing, opposite of each other. So one pair of contrasting traits is called a monohybrid cross. That is to demonstrate the dominance of traits or expression of traits. Whereas second experiment is referred to independent inheritance of traits. The second experiment is called, or it is to demonstrate the independent inheritance of traits, which is a dihybrid experiments. As the name suggests, dihybrid means this experiment is crossing of two visible contrasting pairs of characters. Two pairs of contrasting visible characters. An experiment that involves two pairs of contrasting characters. For example, I can club round wrinkled with color that is green yellow. Green and yellow forms one contrasting pair, whereas round and wrinkled forms, seeds I am talking about, round and wrinkled also forms one contrasting pair. So when I compare these two contrasting pairs, then that experiment, when we compare these two pairs of contrasting characters and when we cross these two, then we say it is a dihybrid experiment. And remember, Monohybrid experiment is to demonstrate the dominance of traits or expression of the traits, whereas the independent inheritance of traits was demonstrated by dihybrid experiment. So here it is written clearly, dihybrid experiment, the cross between two pea plants differing in two pairs of contrasting traits is called a dihybrid cross. So I hope you have understood the monohybrid and dihybrid experiment. Monohybrid, comparison of only one pair of contrasting traits, whereas dihybrid means comparison of or the difference in two pairs of traits is called a dihybrid cross. One pair of traits and two pairs of traits, but contrasting in nature. That you should keep it in mind. Now, a couple of things are there here. What is an allele? Allele means, say for example, if a particular trait tallness is represented by capital T and capital T, in that case each individual T is called an allele. That means allele is a variant form of a gene, or more simple, in the more simple terms, one member of a pair of gene that decides a particular trait or a character. Tallness is a character represented by, say, capital T, capital T. This is a pair of genes or a pair of factors that determines a particular trait, tallness. In that T and T, if I consider only one member, that is one T, that is called an allele. So a gene set or one pair of series of genes or one pair of genes that contains copies, two copies are called alleles. So one member of a pair of gene that determines a particular trait is called an allele. What is a phenotype? Phenotype means observable trait or character of a gene. That means if the gene is expressed, say for example tallness, and if the plants are tall, then we say the phenotype is the tallness. So phenotype refers to the visible character or the observable character of a particular gene. What is a genotype? Genotype is the genetic combination, the organism, what genes it is made up of, what combination of genes it is made up of. So the genetic combination or a genetic composition of an organism is called a genotype. So you should remember these things. Alleles means the variant form of a gene or one member of a pair of genes which determines a particular trait. You can say that. So I hope you have understood here very important things. Mendel's experiments. The monohybrid experiment talks about the dominance of traits or expression of traits, and the dihybrid experiment is meant for showing independent inheritance of traits. How the traits are independently inherited, the tallness or shortness, how those are inherited independently. So we look at those experiments, then it will be much clearer. So let us talk about Mendel's first experiment, which is a monohybrid experiment, and monohybrid experiment is to show that the expression of traits, how it is done, or the dominance of traits is demonstrated by monohybrid experiment. See here, this is the description part, and here you can see here this one. This is a square which have been drawn over here. These are called Punnett squares. These are called Punnett squares. Let me little bit talk about this Punnett square here. In this Punnett square, on the top side we consider the gametes, on the left side we consider the gametes. Here you can say T and T, these are the gametes, and small t and small t, these are also gametes. Generally on the top side we take male gametes, and on the left hand side we consider female gametes. This is in general, okay, but you can simply write here gametes and here also gametes. That is enough over here. So let us see Mendel's monohybrid experiment. What did he do basically? Mendel selected pea plants having one pair of contrasting characters, pure tall and pure short, and he cross-pollinated them. He considered two plants. Pure tall is represented by capital T, and pure short is represented by small t and t. And these two plants are cross-pollinated. In the F1 progeny, when these two plants were cross-pollinated, in the F1 progeny means offsprings. So in the first generation, F1 means generation 1. In F1 progeny or in the first generation, he obtained all the tall plants. What happened to the shortness character? It's a question mark, isn't it? So when Mendel cross-pollinated pure tall and pure short plants, he obtained all tall plants in the first generation. Now, when the first generation plants were self-pollinated, which are all tall plants were self-pollinated, he obtained a mixture of tall plants and short plants. What an interesting fact. When he cross-pollinated the pure tall and pure short, in the first generation he obtained all tall plants. When the tall plants were self-pollinated, in the second generation he obtained a mixture of both tall plants and short plants, but in a one particular ratio. One quarter of the total plants, or 25% of the total plants produced in F2 generation were short plants, whereas 3/4 or 3/4 of the plants were, or 75% of the plants were tall plants. So he concluded that in first generation F1 progeny, both tallness and shortness traits were inherited, but only tallness got expressed. Shortness got depressed or recessed. Recessive trait, you can say. The tallness got expressed. So let me read all these things and then I go back to the Punnett square once again. In figure, tall plants are T, 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 T and T. Short plants are only small t and t. This is how it is, a Punnett square is prepared. So let us look at this Punnett square, how it is prepared. Cross-pollination of tall plant and short plant. He cross-pollinated. Let us take here the tall plant. The genes are represented by T and T. These are called alleles, but the gametes contain only half the number of genes. So only the male sex cells are, or you can say the gametes contain only half the number of these genes here. So male sex cells or the gametes contain T and T whole. There are two gametes here, and here the short plant I will consider, and the gametes of the short plant are small t and small t. So the crossing of this T, one combination, one gamete and gamete of one plant, tall plant, and gamete of short plant put together T and T. Here it is T and T, this T and small t, and this is T and small t. So if you look at this combination over here, when tall plant and short plant are combined together, everywhere you find capital T, small t, capital T, small t, capital T, small t, capital T, small t, and all these plants, 4 plants, were tall plants. No short plants were observed when tall plant and short plant, that is the purely bred tall plant and a purely bred short plant, were cross-pollinated. All the plants in F1 progeny are tall. What is the genetic combination? The genetic combination of these tall plants is capital T and T. So I can say the hybrid of tall plant and short plant led to a genetic combination of capital T and small t. So now can I consider all these tall plants have a genetic combination of capital T and small t? Yes. Now, so in the figure, tall plants are this and this. He observed that 3/4 or 1 out of 4, 3 out of 4 were tall, and sorry, this is the first one. All the plants are tall plants in this. Now he carried out a self-pollination. He carried out the self-pollination of the F1 generation plants. Self-pollination, that is a plant consisting of a genetic combination of TT versus TT. One plant you can take this side, another plant you can take this side. The alleles or the gametes contain one capital T and one small t. This is a capital T gamete and this is a small t gamete. Here also another plant, capital T gamete and small t gamete. Combine these things, T and T, T and T. First let us write capital T and then small t. Then here it is capital T and small t, then small t and small t. Is it right? In the first generation F1 progeny, all these four plants were tall plants, and in the second generation we Mendel self-pollinated these plants having a genetic combination of TT. We get the result as capital T, capital T, capital T, small t, capital T, small t, small t, small t. In this second generation F2 progeny, it was observed that only 25% of the plants were shorter, whereas 3/4 of the plants were taller. That means he finally concluded that only one capital T is enough. Only one copy of capital T is enough to make the plants taller. See here, of course this is a tall plant, and here it is tall plant, here it is tall plant, whereas for a plant to be a shorter, both small t and small t should be present. That means what did he conclude is, from this experiment, Mendel concluded that a single copy of capital T is enough to make the plant tall, whereas both copies to be small t for short plant. That means for a short plant, both the alleles should be identical but only small t. In the second generation, hence traits like capital T are called dominant traits that got expressed in F1 progeny, and traits like small t are called recessive traits that got suppressed in F1 progeny, isn't it? For example, in F1 progeny, this is the genetic combination whereas all these plants are tall plants. Can you find any short plant here in F1 progeny? Not at all. But in F2 progeny, the shortness again came into picture, that is only 25%. That is, out of 4 plants, only one plant was a short plant, whereas all 3 were the tall plants. Therefore, he concluded that one capital T is enough to make the plant tall, whereas for the plant to be a shorter plant, both the alleles should be identical, but it should be a small t. Therefore, Mendel concluded that a single copy of capital T is enough to make the plant tall, whereas both copies of the alleles to be small t for short plant. Hence, traits like capital T are called dominant traits that got expressed in F1 progeny, and traits like small t are called recessive traits that got suppressed in F1 progeny. Now let us take a little bit a ratio of this one, a ratio in this two Punnett squares. One is the phenotypic ratio and genotypic ratio, or phenotypic ratio and genotypic ratio. In F2 progeny, the phenotypic ratio refers to the ratio of observable traits. What are observable traits over here? 3 tall plants, 1 short plant. Observable traits are tall plant versus short plants. The ratio of tall plants to short plant is 3:1. Here you can see, this is tall, this is tall, this is tall, and this is short, 3:1. Genotypic ratio. Can you check the combination of the genes here? This is one particular gene combination, these two another one particular combination, and this is another one particular combination. This is pure tall, this is pure short, and this is hybrid plants, hybrid tall, hybrid tall. Got it? So pure tall to hybrid tall to short ratio, that is genotypic ratio, that is the genetic composition ratio, capital T T is to capital T small t is to small t is to t is 1 is to 2 is to 1. Is it so? 1 is to 2 is to 1. So what is the purpose of this experiment basically? Very quickly, Mendel selected pea plants having one pair of contrasting characters, pure tall and pure short. When he cross-pollinated them, in the first progeny, he obtained all tall plants. In the second progeny, that means when the first progeny plants were self-pollinated, in the second progeny or in the second generation, he obtained a combination of tall plants and short plants. In what ratio? 3 is to 1. 75% were tall plants, 25% were short plants. And therefore, he concluded that in the F1 progeny, only one capital T is enough to make the plant tall, whereas in the second progeny, if you look at, to make the plant short, you require both small t. So, capital T that got dominated in the first generation is called a dominant trait, and so the shortness got suppressed in the first progeny, whereas it got expressed in the second generation. The shortness got expressed in the second generation. So finally, what is the conclusion over here? The last sentence here, very important. What is the conclusion? This led Mendel to propose that two copies of a factor or two copies of genes are the ones that control the traits which are present in sexually reproducing organism. That means in sexually reproducing organisms, one pair or two copies of genes are required to control the traits. That is the biggest discovery. To control any one particular trait, the requirement is two copies of genes, which are called pair of alleles. So this led Mendel to propose that two copies of factor or genes controlling traits are present in sexually reproducing organism, one from the father and one from the mother, and these two may be identical if the two genes are identical. If the two alleles are identical, we call homozygous, or maybe different, then we call heterozygous, depending on the parentage. So this is about the Mendel's monohybrid experiment, and this is a Punnett square. How to prepare the Punnett square? Here you can take tall plant over here, short plant over here. These are the gametes of tall plants, gametes of short plant combined together to obtain all tall plants in F1 generation. Self-pollinate to obtain the F2 progeny. Self-pollination of TT. That means all these plants are having a genetic combination of T, capital T and small t. So these are the gametes basically. Gametes containing capital T, gamete containing small t, capital T, small t. Combine this one. So 3/4 tall and 25% or 1/4 short plants. This is how the Mendel's experiment was concluded. This is to demonstrate the expression of traits. I hope you are able to understand this monohybrid experiment. Here you can see the diagrammatically. I think in the next slide the diagrammatically I have represented. Let me check that one. Here, yeah. Monohybrid experiment, cross-pollination of tall plant and short plant. Tall plant is represented by capital and capital T. See here, generally the dominant traits are, now you know that the tallness was dominant in the F1 generation. So generally we represent the dominant traits by capital letters, uppercase letters, and the recessive traits by small letters or lowercase letters. So tall plant, if I represent it as a capital T and T, and short by T and T, in the F1 progeny, after cross-pollinating these two plants, in F1 progeny, we get all tall plants. Now, let this plant undergo a self-pollination. That means capital T, small t. These are hybrid plants. These are pure. This is a pure plant, purely bred plant, purely bred plant. breed bred, that is the past tense of breed. So all these are tall plants or hybrid plants. When this is a cross pollinated, then 75% were tall and 25% were short. Here we did not find any short plant at all, and of course there was no mixture of medium plants also. All the plants were tall, so the tallness got expressed in F1 progeny and the shortness got recessed or depressed, you can say, and therefore the dominant trait is capital T. Here only capital T is enough to make the plants tall here, whereas for a shorter plant definitely both T should be a smaller, is what Mendel concluded. This is one particular experiment. Here you can see the color of the petals of the flowers, purple and white. When these two are pollinated, cross pollinated, or a cross fertilization takes, when the cross fertilization takes place, then if purple is the dominant color, then in first progeny all the plants will be purple color. Once this purple colored plants are self-pollinated, the offsprings of the second generation will have this combination: 3/4 purple, 1/4 white. Similarly, if you compare here green colored seeds, but one pair of contrasting traits are the shape, round and wrinkled. Let us assume that round is a dominant here, represented by capital R, and wrinkled small r. In the first generation, if you cross these two and obtain a Punnett square, you will find RR combination. Capital R and small r combination will be there for all four plants. So capital R is a dominant trait, and in F1 generation all these are round seeds are produced. When the self-pollination of F1 plants are done, you can find the mixture of round seeds as well as wrinkled seeds. But what is the ratio? The round seeds is to wrinkled seeds ratio is 3:1, and this is a phenotypic ratio. I hope you understand. So I have done a few more Punnett squares for a few more traits here. Monohybrid experiments, tall and short plant with respect to height. The contrasting visible traits, these are the contrasting visible traits. Tall and shortness is one particular trait, round wrinkled, yellow green, purple white petals, and these are seeds, petals, height. So height, shape, color, these are the contrasting traits which have been taken and prepared here. So for example, this one which here we are comparing the height here, trait height, cross pollination of tall plant and short plant represented by like this. Alleles present in the tall plant, let them be TT. In short plant, T and T. One from father, one from mother. One from father, one from mother. So tall plant gametes are represented here. Short plant gametes are represented by here. Gametes have half the number of genes. If this tall plant contains a pair of genes in its sex cell or in its gamete, only one T can be observed. So there are two gametes with T and T. Here also there are two gametes. If you combine these two, you can get in F1 progeny all these are tall plants. Therefore, expressive trait is capital T, tall, and recessive trait is small t, short. Self-pollinate the F1 plants. Alleles are the hybrid alleles. The genetic composition is capital T, small t, cross capital T, small t. This is a self-pollination. So capital T, small t for one plant, capital T, small t for another plant. Combine these together, you get this is the genetic combination. Phenotype is 3 tall plants, 1 short plant, and therefore phenotypic ratio is tall is to short is 3:1. Genotypic ratio, the ratio of the genetic combination of the plants, capital T and T is to small, capital T and small t is to small t is to small t. This is 1, these two are 2, and this is 1. So 1 is to 2 is to 1 is the genotypic ratio. I hope you understand this very clearly. Next. Yeah, here I have considered trait is shape of the seed, cross pollination. The shape of the seed is here round and wrinkled seeds. If we represent round as capital R and R, that means this is a dominant trait, whereas wrinkled, let us consider a recessive trait. In F1 progeny, this is the combination. How did you get this one? The gametes of plants containing round seeds are represented here. Gametes of plants containing wrinkled seeds. Combine them, RR. All these are all round seeds because only single R is enough to make all the seeds or the plant containing all the seeds round. So all round seeds are produced in F1 progeny. Self-pollinate these plants. F1 cross F1 means self-pollination of F1 progeny plants. These are the alleles, gametes, combine it. F2 progeny result is RR is a round seed, RR is also a round seed, small r refers to wrinkled seeds. So phenotypic ratio round is to wrinkled is 3 is to 1, and genotypic ratio this is to this is to this is 1 is to 2 is to 1. Similarly, let us compare the petal color. Petal color, cross pollination of purple and white. Let purple be capital PP, white be small p. In F1 progeny, gametes of purple color, gametes of white color. This is the combination. Capital PP is the genetic combination. All purple petal plants self-pollinate this plant. That means capital P small p cross capital P small p. Gametes contain capital P small p. Here also capital P small p. Combine this. The phenotypic ratio, that is purple is to white. Purple, see here before that, this one and this one and this one. This is purple color because capital P is present. Small p, this is white. So phenotypic ratio purple is to white is 3 is to 1. Genotypic ratio is PP is to capital P small p is to small p 1 is to 2 is to 1. This is a genotypic ratio. So finally, very important one conclusion before we conclude. I think here it's the end of the lecture. So in my next lecture we'll be taking the dihybrid experiment, but before we end the lecture over here, very important one thing, that is here this observation is most important. So after all, what is the conclusion of the Mendel's monohybrid experiment? Conclusion of the monohybrid experiment is that this part and the last part, these are very important. Mendel concluded that a single copy of capital T is enough to make the plant tall, whereas both copies to be small t and t for short plant. Hence, traits like a capital T are called dominant traits, and traits like small t are called recessive traits. This is one part. And second thing is, here this experiment led Mendel to propose that 2 copies of factor or 2 copies of genes or 1 pair of genes are required to control a one particular trait in sexually reproducing organisms. I hope you understood the Mendel's monohybrid experiments and its conclusion. So let us end the lecture over here. In my next class, I'll be taking the dihybrid experiment, which is to demonstrate the independent inheritance of traits. So thank you for watching. See you in the next lecture.